Okay, welcome to Chapter 9, where we're going to be talking about viruses and cancer. And I've got to have this split into two parts. In the first part, I will talk about defining cancer, some of the properties of cancerous cells. We'll discuss just basic cell cycle review and control of the cell cycle, including checkpoints, cyclins, and CDKs. And then I will mention two main groups of genes, oncogenes and tumor suppressor genes, that are key to development of cancer, and I'll give you an example of each. In the second part of the lecture, we will go through some of the examples of known cancer-causing viruses. All right, so obviously cancer is a huge issue, a huge medical problem. So just in the United States, over 1.5 million people were diagnosed with cancer just in the year 2015. That number has not gone down. If anything, it's increased in the last 5 years. If you are a man in the United States, you have a 50-50 shot of developing cancer in your lifetime. If you're a woman, you have a 1 in 3 chance. So cancer accounts for about 1 out of every 4 deaths. 1,600 people die of cancer every day, over 500,000 people every year in the US, and globally we're talking over 8 million deaths. So obviously there's a lot of interest in understanding cancer and trying to come up with ways to treat or prevent cancer. And it is estimated that viruses cause about 15% of cancer cases worldwide, most of those being either cervical cancer, liver cancer, or some oral cancers. But that's really significant, because if you think about, okay, these are cancer cases being caused by infectious agents, if we could prevent exposure to those infectious agents, we might be able to eliminate 15% of 8 million cancer deaths, which would be a huge deal. So what is cancer? Well, simple definition would be it's uncontrolled cell division that is going to lead to these cancerous cells invading nearby tissues, potentially even spreading to distant parts of the body. But anytime you have too much cell division, you're going to develop a tumor, whether that be benign or malignant. And there's a number of steps that a normally dividing cell would have to go through to cause tumor formation. The first of which would be mutation in probably multiple genes in that particular cell. And we'll talk about this a little bit, but it's these changes in the genetics of the cell that make a normal, well-behaved cell become cancerous. Second major step is what's called angiogenesis, which is where tumors can actually recruit their own blood supply. That allows for them to gain extra nutrients, discard waste, and it'll allow a tumor to grow very quickly. And then the third, of course, is metastasis, which is the migration of those cancer cells from the site of the original tumor. And this is what makes cancer really so deadly, right, is the fact that it can spread to multiple parts of the body, establish secondary tumors, and that makes it very difficult to treat in most cases. So how do we go from a normal cell to a cancerous cell? Well, one well-known way of doing that is by exposure to things called carcinogens, which are just substances that induce changes or mutations in the DNA. And these can be categorized into what are considered physical carcinogens. That'll be things like UV light, or high energy ionizing radiation, X-rays, or things like that, versus chemical carcinogens. So it'd be something like asbestos, or of course there's a long list of known chemical carcinogens in cigarette smoke, for example. And then you have biological carcinogens, which would include viruses, bacteria, parasites. As we're going to see, there's quite a few viruses that have been identified as biological carcinogens. There's just a few bacteria and only a couple of parasites that fall into that category. But obviously our focus here will be on the viruses. Now this change from from normal cell to cancer cell is often referred to scientifically or medically as transformation, this transformation process. So this would include all the physical phenotypic changes in those cells. And of course, those physical changes are a result of the genetic changes, the mutations that I already mentioned. So, what are the phenotypic changes? Well, of course, we're talking about cells that are dividing too much. So we've dramatically increased the rate of cell division, proliferation. Oftentimes these cells become immortal, meaning they can divide indefinitely, which normal cells cannot do. And they change their shape in a number of different ways. But just to give you one example, you can see some normal nasopharyngeal epithelial cells here on the left, and then some transformed nasopharyngeal epithelial cells on the right. And you can see they actually have a visibly different shape, right? So their morphology has changed due to these genetic alterations. So other components of transformation, we talked about immortality. One way cells become immortal is they can reactivate this enzyme called telomerase. So one of the factors that usually limits how many times a cell can go through division is the length of the telomeres, or the ends of the chromosomes. Those shorten with every round of cell division, and eventually they shorten to a point where we get in danger of important genes and genetic information being affected, and then that cell would normally go through programmed cell death or apoptosis. But there is an enzyme called telomerase that can regrow telomeres, and normally that enzyme would only be produced in things like stem cells that have to divide a very large number of times over a long period of time. But cancer cells are very good at activating that enzyme, and that's thought to contribute to their immortality. A lot of cancer cells can actually produce their own growth factors, so they're not relying on external growth factors, these signals to stimulate their growth. They will lose what's called contact inhibition, so normal cells don't like to be piled up on top of each other, right? They'll stop dividing if they run out of space. Cancer cells don't do that. They also they acquire what's called anchorage independence. Normal cells need to be anchored on a substrate, so petri dish, or in a human body, right? Extracellular matrix or another layer of cells in order to grow well. Whereas cancer cells lose that dependence, they're happy to grow in circulation in the bloodstream, which most normal cells do not do, or cannot tolerate. And I'm not going to go through all of this, but this is a pretty well-known list of these 10 hallmarks of cancer cells, and this was a list put together by these two very famous cancer researchers. And some of these things we've already mentioned, basically increased proliferative signaling, evading things that might suppress growth, cellular immortality, etc., inducing angiogenesis. So again, I won't go through the whole list. You can give it a read on your own, but you're going to find these 10 hallmarks or characteristics in basically every cancer cell that you look at. And again, all of these hallmarks are due to genetic changes that have transformed these normal cells into cancer cells. And so because this is reflected of a large number of genetic changes, it will take years usually for cells to acquire all of these characteristics, and that's one of the reasons that cancers are typically diseases of old age, right? It takes lots of years to get all these mutations in a single cell and convert them from quote-unquote normal to cancer cells. Okay, so hopefully that gives you at least a basic feel for what are some of these key characteristics of cancer cells. And now I want to shift and turn to talking about, well, if if cancer is a disease of too much cell division, where cells are going through the cell cycle too quickly, what does the cell cycle look like, and what normally regulates that process? So if you've taken another biology class, I'm sure you've seen this before, but we have the cell cycle here where M is mitosis, right, cell division, so you could consider the end of mitosis sort of to be the beginning of a new cycle here. This would be cytokinesis here, dividing cell. Then we go through what's called the G1 stage, then into the S phase, and then G2, and then we'd be back to mitosis. So this whole portion of the cell cycle that is not mitosis, G1S, G2, is referred to as interphase. In G1, the cells are growing and metabolizing. S phase is where DNA replication takes place, where we copy the DNA, basically getting prepared for the next division. G2, again, there's more basic metabolism, but we're really ramping up preparation for mitosis. And what I wanted to point out here is you'll see these red boxes, which are some well-studied or called checkpoints in the cell cycle. And so these, you could think of these as red traffic lights or stop signs in the cell cycle, where the cell will typically be forced to stop at these checkpoints, and it's really a chance for the cell to assess, is everything okay? Is there any DNA damage? Have we picked up too many mutations, or have our chromosomes been altered in some way? If not, if everything seems okay, then the cells should proceed past those checkpoints. But it's really kind of a quality control opportunity, essentially. And so what determines whether or not a cell is going to pass those checkpoints is this pair of proteins called cyclin and CDK. So CDK stands for cyclin-dependent kinase. Kinases are just a general class of enzyme whose job is to phosphorylate or add phosphate groups to other proteins. And so the kinase, the CDK, has to pair up with this partner protein, cyclin, so they physically bind to each other. And once that happens, they become activated, and that cyclin CDK now can start phosphorylating inactive proteins in the cell and activating them, or vice versa. Even could take an active protein, phosphorylate it, and inactivate it in certain instances. But basically, just to go back, if everything seems to be okay at these checkpoints, at these stop signs, then cyclin CDK will phosphorylate some protein that will turn that red light green, and the cell can proceed past the checkpoint. And then it'll stop at the next checkpoint, and we would need another cyclin CDK pair to show up and phosphorylate something in order to proceed. So they're really kind of the gatekeepers at these checkpoints, so to speak. And we're not going to go into all the details here, but I do want to point out there's different versions of cyclins and CDKs. You can see just in this example, there's like CDK1 or CDK2, cyclin A, cyclin B, cyclin D, cyclin E. So different combinations of cyclins and CDKs will be working at different checkpoints. And so what we see in cancer cells is often some of the mutations that those cancer cells have acquired may affect the cyclins or the CDKs or other involved regulatory proteins that basically cause this whole system to break down. So the cell is no longer stopping at these checkpoints, and it just blasts through all the red lights, regardless of whether there are too many mutations or some other significant problem with the cell. And certainly cancer-causing viruses, often called oncogenic viruses, can contribute to this. So as we'll see, there are some viruses that actually encode proteins in their genome that may contribute to this loss of cell cycle regulation. All right, the last thing I wanted to cover in this first video is this distinction between oncogenes and tumor suppressor genes. So these are the two big classes of genes that play an important role in cancer development. And oncogenes are genes that promote too much cell division. So you can think of these as genes that are stepping on the gas pedal. Tumor suppressor genes, their normal job would be, as the name implies, to suppress cancer, to step on the brakes and slow down cell cycle progression. And so what happens in the case of oncogenes, these these cancer-causing genes are actually present in your genome in the form of what are called proto-oncogenes. So these are just normal genes whose normal job is to promote cell division, right, under the appropriate circumstances. And what happens is if these proto-oncogenes are mutated such that they gain some activity, we would call it a gain-of-function mutation. Maybe they become constitutively active, or the proteins encoded by those genes become overproduced for a number of reasons. Then that proto-oncogene now becomes an oncogene. And so it's doing its job too well, it's promoting too much cell division, which can obviously contribute to that cancerous state. And then the flip side of that coin, the tumor suppressor genes, would be, as I said, those whose normal job is to step on the brakes and prevent cell division. So if you acquire mutations in tumor suppressor genes that cause them to lose their function, basically break the function of the protein, then you've cut the brakes, right? You no longer have that inhibitory effect. And what you often see in many cancer cells is both of these things are happening simultaneously. So a cell will acquire a gain of function in a proto-oncogene, so that it now has this oncogenic function, too much cell division, and you lose function of one or more tumor suppressor genes, cut the brakes, and that combination together is not good, right? You're definitely going to get drive the tumor state and potentially cancerous state in that instance. And so I just wanted to give you one example of both of these categories of genes. So this is a cell signaling pathway called the RAS pathway, and you don't have to know all the details here. If this is something you're interested in, you could always take my biology of cancer class and we dig into this. But the bottom line here is this signaling pathway is initiated by binding of a growth factor to a receptor on the surface of the cell, and then you get this this whole chain reaction of, one protein activates the next, so on down the line, until the end result of this pathway is we turn on transcription and translation of cyclin CDK, or specific cyclin CDKs, which, as we talked about, then will allow the cell to continue through the cell cycle, right? So this whole pathway is geared to help promote cell division. So it's a proto-oncogenic pathway. And we could consider really any of the genes, any of the proteins in this pathway to be proto-oncogenes. So really, if any of these different proteins that you see on these images acquire mutations that give them too much function, then the end result is probably going to be too much cyclin CDK. So actually this is called the RAS pathway because one of the key components of this is this protein called RAS. It was actually the first oncogene identified. And you see RAS mutations in a very high percentage of cancers. Excuse me. The example I want to show you of a tumor suppressor gene is what's called the retinoblastoma protein. And so retinoblastoma, this protein, which is so named because it was first associated with cancer of the retina, retinoblastoma, it basically sits at one of these cell cycle checkpoints, specifically the G1 to S checkpoint, and it just kind of hangs out there, and what it does is it binds to this transcription factor called E2F, and it prevents that transcription factor protein from going into the nucleus and activating transcription of genes that would then be used to replicate the DNA to go into S phase. And so basically that is the red light, the stop sign, right? Is retinoblastoma preventing this transcription factor from starting that whole DNA synthesis process. Well, what happens if we have a mutation in retinoblastoma, right? A loss of function mutation is there's now no longer anything to inhibit that transcription factor, so it's just always running off to the nucleus to turn on transcription. Normally, that would only happen if we have the correct cyclin CDK pairs show up and phosphorylate retinoblastoma, then it would release the transcription factor. But if retinoblastoma is mutated, then all this is off the table. We don't even need that. We're just always over here. We're always going past that checkpoint. So hopefully that kind of sets the stage for you. And as I said in the second video, I'm going to go over some specific examples of known cancer-causing viruses.